days, school days, dear old golden rule days, reading and writing and arithmetic, taught to the tune of a hickory stick. You were my queen in calico, I was your bashful barefoot beau, and you wrote on my slate, I love you, Joe, when we were a couple of kids.
Hi, welcome back. As you can see, we're exploring another exciting depth to our world, and it can be revealed through the use of the microscope. This is lab number 5, the fungi cell represented by a yeast cell. More specifically, the example of the fungi cell we're using are the yeast cells. Fungi is plural for fungus. Fungus is the singular form. Fungi are decomposers. They are able to recycle and decompose dead plant and animal material. Today we are going to study the yeast cell, but we're going to make our own preparation. As you can see, you need 1 package of active dry yeast from either of your parents, or maybe you cook or a friend cooks. Just get 1 package as seen here. And you need 2 tablespoons of sugar, and 1/2 cup of lukewarm water. And you stir this around in a bowl, and it's frothy, and it looks like dirty tadpole eggs. And it smells like bread. This experiment, you need a lot of patience for it, because there are many cells multiplying right now. To make your prepared slide, get a clean cover slip and a clean slide, and put a very small drop of yeast solution on your slide. Very small drop, like there's going to be thousands of cells in just that little drop I placed in the bowl. Put it on your slide, then put the cover slip on top and fasten it in to your microscope. Begin with 4x objective to magnify. You're going to see thousands of cells. And then to be able to witness what you want to know is the actual budding of the cell, which is a split in 2 equal halves. You need to focus on 400x. As you look through the microscope, you should see a cell budding. One is the mother cell, the one on the left, and the one on the right is the daughter cell. They are going to be identically similar. The nucleus, which is not seen there, is splitting in half. Notice that the mother cell is slightly bigger than the daughter cell. Nucleus is splitting in the center there between the 2 budding cells. Where the split is, that's where you put a dotted line overlapping, like a Venn diagram, overlapping the 2 cells and in equal parts. So, more simply stated, the yeast cells multiply by asexual reproduction, as seen here, by two cells splitting in equal halves. The cell membrane protects the yeast cell from its external environment. The cell wall of the yeast cell is composed of a fibrous material called chitin, whereas the cell wall in the onion cell was covered by a cell wall that contains cellulose. External plasma membrane can be seen in the yeast cell. See the indentation where the two cells are splitting? That will be completed in the cell membrane, and you'll have two cells with their own cell membranes. In the yeast cell, the cell wall is not seen, while in the plant cell or the onion cell, there are chloroplasts. However, the yeast cell do not have chloroplasts, so they don't make their own food. The food for the yeast cell was the sugar that we added in the beginning to the solution of yeast. That is what made it grow so big and multiply so many millions of times. It is time to take out your biology notebook. Label this lab as lab number 5. Write out laboratory number 5, yeast cells. The introduction should contain the purpose. The purpose is to draw and label a unicellular yeast cell budding. Include the materials after the purpose. Write instructions on how to prepare the yeast cell. Record your observations stated in the purpose. At the end, summarize with a conclusion. Remember to clean and store your microscope. Use the 4x objective as the one in place, and make sure you've used your lens paper to clean any wet surfaces and objectives and the ocular lens. Turn off the light. See you next lab.